Welcome, everybody, to this H3D Foundation webinar. I'm Nicola Elliott Wang, and it's my great pleasure to host these sessions for the Foundation. Today, we're really excited to have Professor David Fidock join us to speak about, especially in response to a call from our malaria team, from the GC ADA malaria team. So I'm going to kick us off by reading his bio. I think he's well known to many of you, but it's always good to get the nitty-gritties from the bio to give us more nuance. David Fidock is a professor of microbiology and immunology and of medical sciences at Columbia University in New York, and the scientific advisor to the president at Institut Pasteur in Paris. He graduated from the University of Adelaide in South Australia and completed his PhD in microbiology at the Institut Pasteur. His research focuses on the genetic and molecular basis of Plasmodium falciparum resistance to first-line antimalarial drugs. He also investigates resistance risks and modes of action of compounds in the antimalarial drug discovery and development pipeline. He is immediate past president of the American Society of Tropical Medicine and Hygiene, ASTMH, and serves on the WHO Malaria Policy Advisory Group. He has received the Bailey K. Ashford Medal and the Trager Medal from the ASTMH, a Global Australian of the Year Award in Life Sciences, and the Alice and C.C. Wang Award in Molecular Parasitology from the American Society of Biochemistry and Molecular Biology. He is a fellow of the American Academy of Microbiology.
Thank you, Nicola, and thank you everyone for joining today. It's an honor and a pleasure to present to this H3D Foundation. I've worked with Kelly Chibali for many years, and actually a number of his trainees have joined my group over the years. Catherine Wicht, John Okombo, more recently and presently, Lan Elgani and CJ Wele are both postdocs in my team. So it's really been great over the years to work with UCT and with H3D. We have ongoing, continuing collaborations with Lauren and Arence and Catherine Wicht and colleagues there, and also other colleagues in South Africa for our work. So as Nicola said, so I sort of hold two affiliations since 2008. 2007, I've been at Columbia University in New York, in Manhattan. And then starting last September, I sort of rejoined Pasteur in a part-time role where I advised the president and help with international relations, primarily, basically. So without further ado, I'll jump into the topic. So my focus of research, I guess, since about 1988 has been on malaria. So this is just some of the salient points. As we know, malaria continues to exert a massive burden of disease, over 280 million cases in 2024, over 600,000 deaths, mostly in young African children in sub-Saharan Africa. And we know that of the approximately 120 Plasmodium species, there are 5 that are infectious for humans. I'll be talking today about falciparum, which is the most lethal, the most problematic in sub-Saharan Africa, death occurring by a number of mechanisms, including severe anemia, multi-organ failure, cerebral malaria leading to herniation and respiratory distress or arrest. And there are other Plasmodium species as well in sub-Saharan Africa, Ovale, Nolzi. Well, actually, Nolzi is not in Africa. Malaria and Vivax in the Horn of Africa, which has the issue of relapsing malaria. So these are all transmitted by Anopheles gambiae being the major species of concern in sub-Saharan Africa, although later I'll talk about Stephen's eye. And so when we look at the life cycle, so it's important to know then the infection in the human host begins with a very small inoculum, fewer than 100 parasites delivered by a mosquito bite, coming into the skin, finding their way to the liver, invading hepatocytes, and taking advantage of this very metabolically rich environment in the hepatocyte to replicate prodigiously within about 7 days. In the case of falciparum, there's the production within a single infected cell of about 10,000 infectious parasites. These will then exit from the liver. They shroud themselves in hepatocyte membrane to sort of hide themselves away from the phagocytic response. They then have a number of receptors on their surface that allow them to recognize and invade erythrocytes. So these are mature anucleated red cells. They then initiate the asexual blood stage of development. This is about a 48-hour period. And morphologically, we distinguish these as rings. These early forms here, trophozoites, which are more metabolically active, DNA starting to replicate, parasites then segregating during this process of schizogony into merozoites. A much smaller proliferative cycle in terms of numbers, anywhere between 8 and 32 parasites per infected red cell. And these parasites will then exit from the infected red cell, recognize and reinvade another host red cell in a process that lasts about 30 seconds. So there's a very brief period of extracellular appearance of these parasites in the circulation. Just to point out also, these rings are the circulating form. As the parasites mature and change their membrane and become easier to recognize by the spleen, they will sequester in the microvasculature using parasite proteins that they put onto the surface of the infected red cell to basically remove themselves from the circulation and prevent splenic clearance. So this is the asexual stage. This is relatively easy to culture. And so we invest most of our energy on this stage because this is the stage that's therapeutically the most relevant for falciparum. There's also the sexual stage of development. These are called gametocytes. They have males and females. And this requires about a 2-week period before they become mature stage 5 gametocytes. That can then be taken up during a blood meal with exflagellation of the males, fusion with the females, formation of these okinetes, sexual recombination occurring at this stage, leading to the production of these sporozoites in this oocyst that then complete the life cycle. So the human host is all haploid, and that makes it much easier when we're dealing with an organism of about 5,300 genes. You only have to target one copy of each gene. This is a schematic representation of the infected erythrocyte. And so the parasite itself is separated from the red cell by 2 membranes, the vacuole and the parastophorous membrane. And within the parasite, as the parasite matures, there's also an increased volume of this digestive vacuole, which is a very acidic compartment within which hemoglobin is degraded and broken down to provide a source of peptides and amino acids for protein synthesis. So this is a central feature of development, an important therapeutic target. So it begins then with the endocytosis of hemoglobin as from the membrane, basically capturing host cell cytosol, importing it into the DV. Globin is separated from the heme. The heme is highly reactive. It will rapidly oxidize in this very low pH environment. And the globin is then broken down through these series of enzymes, including plasmepsins here, which I'll come back to later, into ultimately amino acids. Now, this process of heme liberation, as I said, is highly reactive. In order to detoxify this very reactive species, the parasites will then form a hemeton, a beta hemeton dimer, that's then the building block for the formation of hemosome crystals, which are essentially lattices of beta hemeton that are chemically inert. And many of the antimalarial drugs used as first lines actually interfere with this process of heme detoxification. So they bind the heme or the actively growing surface of hemozoin and prevent further detoxification. This is therefore a mechanism that the parasite can't mutate the target because the target is of host origin. And the way it achieves resistance is to modify transporters, solute transporters, and convert them into transporters that are able to recognize and translocate drug. And that's what I'll show here with PFCRT a little bit later, that through a series of mutations transforms from a peptide transporter to a drug peptide transporter capable of translocating drugs out of the DV. So it's a long and complex history of antimalarial chemotherapy. I didn't show quinine on this slide, but quinine predates all of this. back to about the 1600s. But here I'm beginning then with the timescale of the various antimalarials used as first-line drugs, beginning with chloroquine in the mid-40s, which was the foundation of a major push to try to eliminate malaria from many parts of the world, not from sub-Saharan Africa. And in these inverted black triangles is the year when clinical resistance or treatment failure was sort of first, convincingly reported. And so you can see that chloroquine had about a decade before resistance was first documented, and I'll come back to that a little bit later. Sulfadoxine pyrimethamine is an antifolate combination, and you can see within a year already, this combination had encountered resistance and continues to do so. SP, as we call it, or Fansidar, continues to be used for chemo prevention, but is done so in the face of abundant resistance. And you can see then here there was a period when really there was very little that was being used that was effective. The monotherapies that were tested for as alternatives were succumbing to resistance. And so we were faced with a period in the 80s and 90s where malaria rates skyrocketed up to nearly 1 million deaths per year as chloroquine resistance and SP resistance migrated across the continent from east to west. So the breakthrough came then with the identification of the chemical structure of artemisinin, a very potent antimalarial that I'll come back to later, which is really the cornerstone of all the first-line artemisinin-based combination therapies widely used as the first-line drug worldwide. And there are several versions of this. One is called AL. It's a Novartis drug. It's called Coartem. This is a combination of an artemisinin derivative called artemether with lumefantrine. And as you can see, and we'll talk about this later, there's no evidence of treatment failure with AL that's conclusively associated with the gain of parasite resistance. So far AL is retaining efficacy. The concern is whether that will ultimately fail. ASAQ, this is amidiaquine. This is the second most widely used first-line therapy across the continent. There are instances of resistance to ASAQ. DHA proparaquine, I'll focus on this extensively later. This was a very important first-line drug in Southeast Asia that failed, as did artesunate mefloquine, ASSP. This is a new one. This is artesunate pyronaridine. It's quite expensive. There were some safety concerns. They don't seem, it seems that actually it is safe. It hasn't yet been widely adopted across Africa, but is an interesting alternative combination. And on top here, what we're showing are these mutations in this gene that's known to be the primary determinant of parasite resistance to artemisinins, occurring first in Southeast Asia, and then more recently being identified in parts of Africa and increasingly seen in regions across the continent. So I want to focus first on chloroquine. This was for many years the first-line drug, has been replaced by artemisinin-based combination therapies or ACTs. But we've learned a lot about mechanisms of antimalarial resistance by studying chloroquine. Shown here then is the schematic representation of the transporter that's primarily responsible for resistance to chloroquine. This gene was identified through a genetic cross between a resistant and a sensitive parasite that differed at 8-point mutations. And if you look at sort of the plethora of mutations that have been identified in this gene from different parts of the world, you can see with this 10 transmembrane protein, many of these mutations within the transmembrane domains. And I'll come back to a more structural analysis of that a little bit later on. If you look here on the right, you're seeing all these different residues that are variant across different geographic regions of the world. The first row is the wild type sequence. And then you can see here chloroquine-resistant parasites from Asia and Africa. You can see a lot of similarity, probably very similar originating events. And then a distinct haplotype common to the Western Pacific and to South America as well. And in gray, then are all these different point mutations that distinguish them, that are separate from the wild type allele. What is common is this one amino acid substitution at position 76, it's K76T. And this has been, and this mutation is present irrespective of the origin and the haplotype of this gene in different parts of the world. And this has proved to be a very effective molecular marker because a PCR assay was developed that's relatively simple, where you can just amplify this piece of the gene. There's a change of a restriction site, and you look for a change in the fragment band pattern, and you can then tell if this is a chloroquine-resistant parasite. And this is associated with a much higher risk of treatment failure, about a 15-fold treatment failure. It is a function of age as well, but it was very apparent when we looked at this across Africa, when was this identified in 2000, that actually parasites were almost always resistant and so there was a switch to at the time ACTs. So as I said, ACTs took over in the early 2000s. These are semi-synthetic derivatives of a product from Chinese sweet wormwood. So it's a natural product to begin with, very potent. So you can imagine if a host has, let's say, 10 to the 13 red blood cells, you could have up to 10% of all your red cells infected by the parasite. This drug is so potent that it's reducing the parasite biomass by up to 10,000-fold every 48 hours. So very rapid onset of action. It also kills the early gametocytes, so it does help reduce transmission. Disadvantage initially was because of the complex sourcing of the material and the need to do semi-synthetic derivatives. It was initially very expensive, but Novartis has been able to get that down, as well as many of the other pharmaceuticals. So now it's much more affordable than it was. They do have very short plasma half-lives, so we're talking about 1 hour often for the artemisinin derivatives. And so this is not enough to eliminate all of the parasites. And so you need these partner drugs to eliminate any residual parasites remaining. So this has to be with combination therapies. Sorry. So this initially was extremely effective, but there were signs already in the early 2000s that there was some clinical resistance starting to arise. And this was a study that was published in 2009 by Dondal et al., where they compared a cohort from Cambodia with another from Thailand, doing a monotherapy trial where they then looked at the parasitemia here shown on the y-axis as a log scale. And you could see this cohort from Thailand very rapidly decreasing their parasite biomass to levels that were essentially undetectable within 2 days. In contrast with this cohort from Cambodia, where you could see a much slower parasite clearance rate, so delayed clearance, with parasites sometimes being detectable still on day 3. And this is called day 3 positivity. So that's a metric of artemisinin, what we call partial resistance. And so this was in Cambodia. It has continued to, there were major efforts underway to identify the gene. Genome-wide association studies was a major approach to try to localize the causal determinant. Ultimately, the breakthrough came from a PhD student in Toulouse who basically took an African parasite and exposed it over multiple cycles over multiple years to increasing concentrations of an artemisinin, and was sequencing the genome as they went and identified a number of mutations, including mutations in a gene which we call K13. The group then, Didier Menard and colleagues, went and looked at parasites in Western Cambodia and found that this particular gene often harbored a point mutation that was associated with this delayed clearance. They also developed an assay called the ring stage survival assay, where you take early ring stage parasites that are very susceptible to artemisinin. These are the stages that that acquire artemisinin partial resistance. And you can do this ring stage survival assay with these very synchronized early post-invasion parasites. You expose them to drug for 6 hours, you wash it away, and then you assess 3 days later the percent survival. And what you can see here In this panel on the right, we had recently just developed gene editing for Plasmodium falciparum. This is a log 10 scale, and you can see just by removing this one amino acid substitution in these two Cambodian artemisinin resistant parasites, you can see a complete loss of resistance. And conversely, if we took a wild type parasite with the wild type K13 allele and you introduce this mutation, you can see a gain of resistance in these two separate genetic backgrounds. So there's sort of definitive genetic evidence that this gene was causal for this artemisinin partial resistance phenotype. So it's an unusual phenotype. It's basically survival in the presence of drug. The parasites cannot proliferate in the presence of drug. And this was shown by Sashil Mock in a study from a few years ago, where she took parasites that were either wild type or mutant at this K13 locus. We looked at 2-point mutations. C580Y is very prevalent. 539T is less prevalent, but very resistant. And you can see this is using microarrays to sort of sample the development of this parasite over a 48-hour period. And you can see here normal proliferation in the absence of drug pressure. If you expose these parasites to DHA, dihydroartemisinin, for 6 hours, the parasites immediately arrest. So transcription and translation cease within an hour of onset of drug action. And so if they're wild type, they have arrested and they ultimately die. If they're mutant and you give them 6 hours of drug, wash it away as if this was within a host. So sort of trying to mimic the pharmacodynamics of the drug. The parasites reinitiate development about a day later, sort of asynchronously, and then accelerate their way through these trophozoite stages, basically catch up as if they'd never been treated. This is only a subset, but it's a very unusual phenotype. We call it, it's essentially a tolerance phenotype. So they can survive in the presence of drug, a subset, and then reinitiate once drug had dropped to subtherapeutic levels. So we know that the clinical evidence came out in 2009. The evidence implicating the gene was in 2014, and the gene was definitively confirmed in 2015. And so with Didier Menard, we were interested in going back to earlier samples and asking when had parasites started to mutate? And this is a study from Western Cambodia looking back to 2001, so shortly after artemisins were introduced. And you can see here in green, that's the population of wild type parasites. And then these other colors show different variants. And the 580Y, the most successful variant, is in red. And you can see already by 2001 that half of the parasite population already was mutated. And what this tells you then is that, we required 15 years approximately to find the gene that was driving this reduced clinical efficacy. And so that's something that we really need to address, because that's obviously too long between when the parasites have come up with a solution and when we've identified the molecular marker. And you can see this sort of spread over different regions at different sort of rates. So for many years, K13 was not identified in Africa until this study in 2020 came out by Didier Menard, who'd sampled genomes from across multiple sites in Africa and identified an origin of mutant K13 in Rwanda. And then through a phylogenomic analysis, they were using whole genome sequence of these samples, they were able to reconstruct a tree, basically. And you can see with this phylogenomic tree that African parasites segregate from Southeast Asian and South American parasites. Within the African cluster, you can see Rwanda, and within Rwanda, you could see an intermingling of mutant and wild type, meaning that these were de novo events that occurred multiple times on these specific African lineages. So this was really the first evidence of mutant K13 originating in Africa. There have been multiple studies since then. Just to highlight a study that came out in 2023, again from Didier Menard's group with whom we worked, where they looked at multiple sites in Eritrea looking at these markers of resistance. Day 3 parasitemia, you can see these districts. These are different colors represent different years, going from 2016 to 2019 in blue. And so you can see some of these districts quickly increasing the day 3 positivity, and mutant K13 starting to pop up quite quickly. And so this is obviously a major problem in the region, but one of several. The other being that Anopheles stevensii has now come into the Horn of Africa, and this is a mosquito that's much better suited to urban environments, and that, of course, poses a significant threat if malaria then transitions more into an urban disease. And the other being that a major, or the major way to detect falciparum infection is through the use of these rapid diagnostic tests that scan for the presence of a falciparum antigen in the blood, basically. And under this selective pressure, parasites in this region have basically been deleting this gene, and so they come up as a false negative. And we know in 2024 that there was a major increase in malaria cases in the Horn of Africa that precipitated a major increase in vector control to try to reduce the burden of disease. So this is the artemisans. That's sort of the current state. I did want to focus on a couple of the other projects in the lab. One important consideration is that artemisans on their own cause this delayed clearance and this in vitro partial resistance, but do not suffice for treatment failure. It's only when the part of the drug fails that the combination fails. And this was shown most clearly in Southeast Asia when artemisinin encountered resistance, and then the partner drug failed, in this case, proparaquine. And again, working with Didier Menard and colleagues, we have investigated this extensively. So there was proparaquine, DHA proparaquine resistance. Once this multi-drug resistance arose, led to almost 90% treatment failures in parts of the Greater Mekong subregion in northern Thailand. And so a spectacular sort of acquisition of multi-drug resistance. The initial studies indicated that there was amplification of these two tandem genes called plasmepsins that are responsible, that engage in this process of hemoglobin degradation and globin degradation in particular. So this was a molecular marker, but we were interested because when we looked at this chloroquine resistance determinant, PFCRT, we noticed during this period when proparaquine resistance was on the rise that this transporter was picking up all these novel mutations in PFCRT. And proparaquine is similar structurally to chloroquine. It's basically a chloroquine dimer with a central linker. So we speculated that maybe these mutations were contributing to proparaquine resistance. So we went ahead and engineered these into parasites and assessed them for resistance to proparaquine as well as fitness. So shown here on the left, this is similar to the ring stage assay for artemisinin. This time it's for proparaquine. It's a log 10 scale, a survival scale. This is log concentration of proparaquine on the y-axis. And you can see here, these are just the control parasites expressing the chloroquine-resistant allele. If you introduce, for example, this one variant that was associated with clinical treatment failure called the 145I, this is extremely resistant to proparaquine. Basically, no matter how much drug you add, these parasites won't die. These were other variants that were also detected in the region, 93S and 218F, and you can see they're significantly less resistant. But if you then study their fitness, what we observed was that this 145I variant was extremely unfit because it really impacted the native function of PFCRT, imposing a significant sort of loss of capacity to efflux peptides, and so really sort of stunting parasite growth, and a much more muted fitness cost with these other variants. In fact, one of these other variants, 93S, having minimal fitness cost, and this actually was observed to have become much more predominant in the area. Now, we can even see this morphologically because this does interfere with the native function of PFCRT. You get this bloating because parasites are unable to properly process globin and export the peptides resulting from globin degradation. We've also observed some interesting findings, one being that when they gain resistance to piperacin, they often lose resistance to chloroquine, the transporter no longer able to translocate both drugs through the PFCRT. And we've also solved the structure of PFCRT using cryo-EM by ligating an antibody to increase its mass and increase its stability. And so this is the cryo-EM derived structure of PFCRT, two different isoforms, one from South America, one from Southeast Asia. And if you rotate these, you can see that these mutations basically line this in the cavity, where the assumption is that they then engage in binding of drug, but more importantly, in a proton-dependent efflux of drug out of the vacuole, away from its heme target, allowing heme detoxification to proceed normally. And these are just the different colors here, the chloroquine resistance mutations in orange and the piperacin resistance mutations in blue. There continues to be evolution of PFCRT across the world. We've looked at the PF8 database of 32,000 genomes. And one we've been particularly interested in is this one called V141L. That's popped up at a low prevalence in Africa. And we've really sort of focused in on that because we know in Africa that the major partner drugs, as I mentioned earlier, are lumefantrine, which is about 3/4 of the therapeutic use, and amidiaquine. And there's a major push by the WHO and others to increase the amount of amidiaquine to reduce the pressure on lumefantrine. And so what we did here was we took a parasite and we engineered in the 4 different variants of mutant PFCRT, the DD2 one, which is in Southeast Asia, and the other 3 in specific to Africa, called GB4, CAM783, and FCB. And when we introduce this rare variant into these parasites, you can see as a small but nonetheless significant increase in IC50 for lumefantrine and to amidiaquine. And this is surprising to us because usually we know that these drugs work in opposing selective pressures on PFCRT. This is the first instance we've seen a variant that actually selects for decreased susceptibility to both at the same time. So we think this is an important molecular marker that we need to survey in the malarial molecular surveillance campaigns ongoing across Africa. And here I just wanted to highlight also some work from John Okombo, who did his PhD and started this postdoc at UCT, working with Tim Egan and Kelly Chibali. He's now a senior program officer at the MMV, Medicine for Malaria Venture, since July, was with my group for many years. And we've done a lot of work on antimalarials in discovery or preclinical development. This is one called Zydus 19489, where John was able to, this was previously considered to be a resistance refractory parasite, or we used to refer to these earlier as irresistible parasites. And we developed, working with Marcus Lee, who's now professor at Dundee, was in the lab before, a hypermutable strain, which we call the Pol Delta strain. And John was able to progressively select for resistance to this compound using this hypermutable strain, and identified a mutation in PFCRT, a novel mutation unrelated to chloroquine and piperacin, that conferred a moderate level of resistance to Zydus. And what's interesting here is that the gain of this mutation led to loss of resistance to chloroquine and hypersensitization to piperacin, presumably because of interference with the efflux of chloroquine and piperacin through PFCRT itself. And what's interesting then is when we expressed this protein and purified it, we can put it into this recombinant system, this proteoliposome synthetic system, and you can use tritiated drug and measure the translocation or the transport of drug through these different variants. So I'm just showing this one variant here called 78 compared to the variant, including the piperacin-resistant isoform. So this is on the left, chloroquine-resistant, on the right, piperacin-resistant. And then here we can look at chloroquine uptake on the left, and you can see you can basically block the transport of chloroquine by co-incubating with piperacin or with this Zydus compound. And similarly, here, this is the transport of piperacin, which you can also block by adding chloroquine. You can also block it with Zydus. And so this to us is a very appealing potential rational combination whereby you add these two drugs, Zydus and say piperacin, and parasites can only acquire resistance by gaining mutations that force them to become susceptible to the other. So you basically lock parasites in a state where they can't acquire multi-drug resistance through mutations in this central efflux transporter. I'm running a little bit behind on time. Just wanted to quickly zoom through this slide, a few slides here on a more recent approach that we've been using in the lab to try to accelerate the time frame to identify genes that are driving resistance, noting this earlier issue I raised that it took 15 years to identify the gene responsible for artemisinin partial resistance. This is using a humanized mouse model with a humanized liver capable of sustaining human red cells. You can then take a multi-drug resistant parasite, mix it with a drug sensitive, take it through the mosquito for genetic recombination to occur, inject these humanized mice. The parasites will then, that are recombinant, will go through the liver into the bloodstream. You can rescue the human red cells and put them back into culture and complete the life cycle. And we've done this now for 2 crosses. Sarshil Mock, who's now has her independent group, has done the same with a recent parasite from Uganda. And you can then clone out these progeny. You can either do it from the bog cultures, or you can subject them to different drug pressures to diversify your population of clones. And this is with the first cross. We looked at artemisinin because we know in the case of Africa that there must be more than one gene involved. Even if K13 is the dominant mediator, it cannot be the sole determinant. There have to be other potentially compensatory mechanisms that enable resistance to the resistant infections to be less unfit and to achieve higher grade resistance. And we've gone ahead and mapped this, and this is the project from CJ, originally from Zimbabwe, but who trained at UCT, identifying K13 as this peak. You can basically just map the susceptibility profiles of your progeny on a genetic map, basically spanning the 14 chromosomes. K13 came up as the number one peak, not surprisingly, and we've been focusing on these 2 other peaks here that we believe may be contributing to the inheritance of resistance, either through compensation or directly augmenting. So he's been working on that. We've also looked at piperacin. As I mentioned earlier, initially these plasmepsin genes were identified as a molecular marker. We came in and showed that you can get very high-grade resistance through these novel mutations in PFCRT. And actually, when we do these analyses, you can pick up both peaks. So you're able to pick up sort of a two-component mechanism of resistance. We know that plasmepsin multi-copy is enough to slightly decrease potency of the compound. You get a major gain of resistance when you add in this mutation PFCRT, and you get even higher grade resistance when you combine the 2. And this very much mirrors what we have seen epidemiologically. This is data from Malika Imwong, looking at parasites from Cambodia over time as they change their first-line regimen to piperacin. You can see this is amplification of PM2 in yellow and novel mutations in PFCRT in this sort of blue color, attaining very high levels over time. And then when piperacin had failed substantially and there was a move back to mefloquine, you can see these mutations starting to quickly fade as wild type parasites overtake the population yet again. So quite closely mirroring what we can observe in vitro with these cultures. We've also looked at quinine. I don't have a lot of time to present this. This is data that's coming out next month in Nature Micro. The paper's already online. We were interested in quinine because it was the former first line for treatment of severe malaria. We've done a genetic cross with quinine. We've mapped it to the genome, and we know that there are 3 components. PFCRT is one of them. There's a component slightly downstream on the same chromosome, and then a component on chromosome 12. And we've had a close look on that second peak of chromosome 7. And we believe that part of this multigenic trait of quinine resistance is a transporter that looks quite similar to PFCRT that we've called DMT1. That's not a primary driver, but that's contributing as a molecular marker that could be useful if ever artemisinin resistance continues to evolve, compromises efficacy to the point that other drugs need to be brought in. This is already sort of anticipating quinine may be adopted as a solution, and we need to have molecular markers on hand. So just to highlight, as I wrap up, that we're also very engaged with the Medicines for Malaria venture. We're the sort of reference laboratory for compounds in their discovery and development phases. Many compounds come to us to assess the resistance liability and identify the drivers of resistance, and from that we gain insights into mode of action. This is a very active area of research in the group, and also an area where we've done a lot of work also with teams from H3D looking at antimalarial compounds. And similarly, this is a consortium that's funded by the Gates Foundation, funded by Elizabeth Winsler, with multiple teams involved. Kelly Chibali and Lauren are part of this team. Lynn Marie Burkholz also does a lot of work here in this space. Candice as well, from H3D. And this is basically aimed at identifying novel targets that can then be used for target-based drug discovery. It can also come from phenotypic hits, but the idea being to sort of develop new chemical matter or new chemical entities that can continue to sustain the antimalarial pipeline to continue to address and mitigate issues of resistance to first-line drugs. So again, another major part of research in the lab. So I just want to finish by acknowledging many individuals in the group. I should have added Candice here from UCT and H3D, just to point out Catherine and Catherine Wick, Lauren Arence, John Woodland, Kelly Chibali. He was my closing keynote speaker at ASTMH last year when we had the meeting in Toronto. Always gives wonderful talks and is an extraordinary scientist as well. Columbia University, just listing a number of colleagues here who've helped and with whom we work very closely over the years. We do a lot of work with the WHO as well, with MMV and with Gates Foundation. And again, just to thank the members of my team, and just to point out again, Larnell and CJ, who I think are having a good time in the lab, and John, who's recently moved, all UCT alum, basically. So with that, I want to thank all of you for your attention and open it up for any questions.